Okay, this short unit's going to be on how scientists manipulate the genes for some benefit. The benefits can eventually cure disease, treat genetic disorders, improve the nutritional values, and improve the nutritional value of food crops, or improve the resistance of food crops to droughts and insect pests. Major tool used by genetic engineers is the restriction enzymes. We can extract these from viruses and bacteria. These enzymes act like scissors that cut a DNA molecule at specific sites. So here's a DNA molecule, and it picks out a place to make a cut, maybe between G and A, guanine and adenine. When it does make that cut, it makes these pieces, these DNA fragment. So what results is the rest of the DNA with a piece broken out of it, and then the DNA fragment right here. And both these things have sticky, what we call sticky ends. They got part of one strand of the DNA is sticking out, and it can stick to other DNA that you decide to put in, that the genetic engineer decides to put in. So maybe he wants to add this piece. Which is foreign DNA to this DNA right here. So that will stick in the place of the piece, that the DNA fragment that was cut out. So here's a picture of it. Here's the restriction enzymes acting like scissors cutting out this piece. Both the fragment and the rest of the DNA has sticky ends. And so you can put another piece of DNA that got cut out of another type of organism with the same restriction enzyme and the same sticky ends, and you can add it to whatever you want. You can add it to the fragment, or you can add it to the DNA that you got the fragment from. Okay, we use bacteria for their little DNA ringlets called plasmids. So here's the nucleus. Here's the circular chromosome of a bacteria, but here's a plasmid. And bacteria have these little accessory rings of DNA. They're very small compared to the chromosome. And what they do is they use those to put in other genes called donor genes. After we've cut both genes, the plasmid and the donor gene, with restriction enzymes. Once the foreign gene has been stuck onto the plasmid, then you put that plasmid back into the bacteria and let that bacteria reproduce asexually, binary fission, and it'll make many, many copies for you of the plasmid. With every time that the bacteria divides, that plasmid will divide with the donor gene inside it. Why would we ever do that? Well, we've done this to produce insulin for diabetics. It's a hormone that diabetics don't make. So what we can do is isolate cells from a healthy human that can produce insulin, and we cut out the insulin gene out of the chromosome that contains it, and with a restriction enzyme, and then we cut a plasmid with the same restriction enzymes, and then we allow the insulin to splice into, by its sticky ends, to splice into the plasmid that's been opened up. And then we insert that plasmid back into the bacteria, and we allow the bacteria to divide over and over and over again. And that bacteria, having that gene inside of it, a human gene to make insulin, will start producing insulin for us. And then we just purify it from the bacteria culture and make it available to diabetics who need insulin. Okay, so here's a term that we need to know. Whenever you have DNA from two different species of organisms combined together by genetic engineering, you have formed what is called recombinant DNA. So the plasmid with the human gene for insulin in it is an example of recombinant DNA. So here we see a pictorial form what I just talked about. Okay, you get a healthy human cell that produces insulin. You locate the chromosome that has the location of the insulin gene. You cut that insulin gene out using restriction enzymes. You do the same thing with the plasmid. Isolate the plasmid from bacteria. You cut the plasmid with restriction enzymes, the same ones that you cut the insulin gene out of the human cell. You mix those together, they will form a recombinant DNA plasmid. Then you insert that back into a bacteria. Whenever a new organism has recombinant DNA in it, is called a transgenic organism. Transgenic is an organism containing recombinant DNA. Recombinant DNA is DNA that has been combined from two different species. Then you take that bacteria, you grow it in a culture, which is just a dish with the nutrients the bacteria needs to live. Let that grow. Then you extract the insulin, because all those cells will be producing insulin because they have the insulin gene. They do this in a big giant tank. They don't just use culture. So they do it on an industrial scale. Then the insulin is purified and given to diabetics. Okay, another way we use DNA technology and the restriction enzymes is for DNA fingerprinting. It has nothing to do with your fingerprints, but your DNA is unique to another person's DNA unless you have a twin, an identical twin, then both of you have identical DNA. So, what's a DNA fingerprint? Well, you take the DNA from a person, a cell from a person, and you extract that DNA and you cut it up with restriction enzyme, a specific type of restriction enzyme. It will break the DNA into fragments, like we said before. Now, each of us has, we'll have similar fragments. We'll have identical fragments for some things, for some parts of the DNA, but we'll have specifically different size fragments because we all have variation in our DNA. So at a crime scene, if they can find some cells left behind, maybe under the fingernails of the victim, or maybe in some blood, or hair, because at the tip of a hair strand, there's a follicle that can contain cells with DNA. So what they do is they take the DNA out of the cells that are found, and they have a way of multiplying it. I'll talk about that. It's called a PCR, polymerase chain reaction. And you can make multiple copies of that. Then you break that DNA up with restriction enzyme into fragments, and then you can spread those fragments out, and they make a pattern, and you can compare that pattern to suspects' patterns. So it can help police, forensic scientists solve crimes. Also useful for showing our hereditary, how closely related we are to other people. Maybe those people from other parts of the world or other past times, by taking DNA from bones. And also for determining how closely related species are evolutionarily. And we'll talk about that later. Okay, let's talk about how you make a fingerprint, a DNA fingerprint. So you take the DNA, you cut it up in a fragment using restriction enzymes. Then we do this process called gel electrophoresis. Okay, I'll show you that in a minute. Here are the DNA fragments are placed into little wells on a gel plate. And then the fragments of different sizes are separated in an electric field. And they're separated based on their size. The smaller ones move faster in the electric field, and the bigger ones move slower. So here's a picture showing how gel electrophoresis works. Right here we have 3 samples of DNA mixed with restriction enzymes. Restriction enzymes have broken it up into pieces, and we squirt a little bit of each of those samples in 3 different wells. Okay, so here's a well, here's a well, here's a well. Okay, this is connected. Then what you do is you turn on the electricity. There's electrical current. This is when you break up the fragments, they're negatively charged because of the phosphate groups. So they will be attracted to the plus side of the field. So there's an electric field. It's like two plates. You turn on electricity, and it'll push the fragments towards the positive. Okay, now the longer fragments are heavier, and they take a longer time to move down the gel. The shorter fragments move faster. So what you do is you let the electricity run until the shortest fragments have almost hit the bottom. Then you stop the electricity. Okay, so you put some chemical tag on the gel plate, and that tag will combine with the DNA and make it show up and be easily seen so that you can take a picture of it. And so here's the fingerprints from the 3 persons. This would be person 1, person 2, person 3. Now you can tell that person 2 and 3 are more closely related than person 1 and 2 or person 3 and 1. Because look, these match up almost exactly. So here's a picture in 3D of the gel, the blue slab of gel, the sample wells. Here's the negative electrode, the positive electrode. Here's the power supply. And it's pretty simple. The fragments move from negative to positive. Okay. And here I'm telling you what it just said. And the smaller size fragments move quicker to the bottom towards the positive, and you turn off the power once those smallest fragments have come close to the bottom of that gel slab. Now, so the pattern of DNA fragments that shows up on this gel electrophoresis plate is unique to every individual. So let's say this is just a sample. This is just a reference DNA that they put in here. Okay, here's 3 people, and here's their DNA was broken up with the same restricted enzymes, and we can see the fragments are different sized for each person, so they're in different locations. Okay, so when you isolate DNA from a few cells, or you need to make many copies of them so that on the gel electrophoresis, the bands will be obvious. If you just have a few cells, DNA, you're not going to have enough to even detect these bands in the electrophoresis. So PCR, polymerase chain reaction, is a technology used to like a copy machine to make more DNA. you only need a little tiny bit of DNA to start. So without getting into too much detail here, how does a PCR, also called a thermal cycler, do this? Well, it heats up and then cools, then heats up, and repeats this over and over again. So basically, you heat up the DNA in the first part of the cycle, and that causes it to denature, separate into single strands, each fragment. Then the solution is cooled. Well, the two strands are separated. And next, the DNA polymerase produces new DNA strands using the base pairing rules and the nucleotides that have been added to the solution. And then the temperature changes again, and the DNA's linked, the two strands linked together by hydrogen bonding. And then the whole thing's repeated again. The whole cycle, over and over. Every time one cycle occurs, one strand forms two DNA strands. Okay, I'm going to insert this slide in right now so I don't forget about it. Since 1990, scientists have been attempting to find the order of the nucleotides in each of our chromosomes. Remember, there's about 3 billion nucleotide pairs in our 23 chromosomes. This task was actually completed in 2023. Not only were the scientists working on the order of the nucleotides in each chromosome, they were trying to figure out what part of the chromosome was what gene. So what chromosome contained the codes for what protein. This work is ongoing. Okay, we've heard about cloning. Cloning is making an exact same organism from a previous organism. So making a twin, identical twin of an organism after that first organism is already developed. So identical twins are clones of each other. There's been a lot of talk about stem cells. There's ethical issues involved, which we'll talk about as well. A stem cell is a special kind of cell that is not differentiated. It hasn't specialized into a specific tissue cell, but it can develop into any kind of specialized tissue cell given the proper environment. Now, we get stem cells from embryos. This is a stage of the embryo. This is not one cell right here. This is a bunch of cells, and it develops 5 to 6 days after fertilization of the egg by the sperm. Okay, now there's a special part of the blastocyst embryo that is called the inner cell mass. And these contain the stem cells, the embryonic stem cells. These are special in the sense, like I just said, they're pluripotent. That term means they can turn into any kind of cell you want them to be. maybe even a tissue or an organ. So what they're trying to do now, scientists, is in their stem cell research is to replace damaged tissues in people that have lost those tissues by disease or trauma. So the process is this. Here's a person up here that needs tissues that he can't produce on his own, like nerve tissues. He needs to replace nerves that were damaged by, let's say, Parkinson's disease. So what's going to happen is the doctors will take his cell, some of his cells, any cell, it could be skin cell, okay? And they're going to take the nucleus out of that cell. Just take this is a 2N cell, somatic cell, not an egg or sperm. And it could be like skin from the patient. Okay, so it's going to be a empty cell. They're going to take the nucleus out of an egg cell. And take the nucleus out of an egg cell of a donor. Okay, unfertilized egg. So that's a haploid cell. So they have the cytoplasm and all the organelles, but not the nucleus. So what they're going to do is put the nucleus of the person's cell into the egg cell with its nucleus removed. So here we have the donors enucleated, that means without its nucleus, plus the patient's nucleus that was taken out. Okay, that is an embryo. It's a 2N cell. And that cell's gonna think it's a zygote, and it will start doing mitosis over and over until it gets to the blastocyst stage. Now, when it's at the blastocyst stage, they'll make this cold and suspend it to grow, so it won't keep dividing. After that, they'll scrape out the stem cells from the blastocyst and implant those into the damaged area of the patient. And when those stem cells experience the environment that they're in, they will specialize into whatever cell that environment tells them to. So if you put them in your brain, they'll turn into nerve cells. Now, I ask you, is this ethical? There's ethical issues here. We Catholics say, believe that no, it's not ethical, even though you're helping a person who's in pain, who's suffering, trying to help him or her get better. All we're doing is producing embryos that could be another individual if you let it grow, and you're just using those individuals as slaves to help another living thing. So there's an ethical issue. You're creating, we say an embryo is a person in the Catholic Church. You're creating people to kill to help another person. Okay, other people say, no, no, no. These are not people. They're embryos. They're not at all people. And there's that whole argument that us Catholics believe that you are a person as soon as you become a zygote. So like I said before, you can get natural cloning that produces identical twins. Okay, so what happens is the zygote will undergo, here's a zygote, after the sperm nuclei is fused with the egg nuclei. The zygote does many mitoses later. There'll be an embryo, and at some stage in the embryo, the embryo splits for some reason. Cells don't stick together, and you'll get identical twins eventually after many mitoses later. Okay, now we actually in 1996 we cloned artificially by doing the method that we talked about on the previous page. You take a skin cell, you take the nucleus from a skin cell, you put it into an egg cell of another animal, and that egg cell is like a zygote, and you allow it to turn into an embryo. You put that embryo into a surrogate mother that you prepped hormonally to carry a child, and that embryo develops into an identical twin to the animal that the nucleus is coming from. But there's issues with that because the vast majority of your tries die, or they develop into embryos with severe mutations. Okay, this process that I described of taking a nucleus out of a somatic cell and transferring it to an empty egg where the nucleus has been removed is called somatic cell nuclear transfer, or SCNT. Now, once to get that, to kickstart the zygote that you produce into an embryo, mitosing into embryo, you got to shock. You got electrically and chemically shock the zygote so that it will grow and multiply. And here's a picture of a guy putting a nucleus into an empty egg cell under the microscope, of course. It's a very fine needle, glass needle. So this shows the huge odds that you're up against and the number of embryos that you allowed to die. I mean, they die along the way to produce one successful cloned sheep. So here's a skin cell. You take the nucleus out, and you transfer it into an empty egg cell. You give it an electrical shock. Now, let's say you made 434 eggs. So you got 434 zygotes after many attempts, many more attempts. Okay, now those will develop into, of those 434 zygotes, only a small percentage of them actually were able to be implanted in a surrogate mother. The other ones died in the attempt. Okay, so there's a big inefficiency problem here. Every time you have an inefficiency event, an individual dies. The zygote dies. Okay, so of those 29 that were actually implanted, only one was successfully cloned. The rest, other 28, died. They were spontaneously aborted, or they grew to be individuals that had severe problems that caused them to die at birth or after birth, soon after birth. So there's an issue there, ethical issue there too. Now, one of my ed puzzles shows this place that makes buku bucks by cloning your pet who dies, and then you give the body over quickly to this company, and for millions of dollars they will clone your dog, your dead dog. But it takes a lot of attempts, and of course they don't talk about all the dogs that they produced that had problems and had to be destroyed. Also, this has been being used by cattle people, because if you have a prize bull and he's getting old, you try to clone it so that you can keep your profits going. Okay, so here we go with what I just said. Great majority of the pregnancies ended in spontaneous abortion because something went on along the way as the fetus was developing. Okay, so if this was a human, if this became available to humans to use, this would be a horrible thing for the mother. It'd be a waste of money in healthcare providers. Also, as I said before, there's serious health handicaps in the abnormal animals produced by cloning. They're really fat with all kinds of diseases that go with obesity. They have early death. Their limbs are distorted. Their immune system and organs are just have problems, etc. So you can see this would not be at all ethical to use, because you feel like having a twin brother as you get old that is exactly a clone of you. Okay, so once again, not to beat a dead horse here, but when an embryo is formed that we know has a risk of dying, a high chance of dying, that is unethical. especially if it's a human. Also, using stem cells, embryonic cells, and to use them to farm off stem cells, and then which kills the embryos you made. That's unethical. If you believe the embryo is a potential person or is a person. Okay, so also cloning, there's so much inefficiency to it. So many embryos that you form die or form damaged individuals, very damaged individuals. That's unethical. And you might say, well, we're doing sheep. We're not going to do a human, but we'll do sheep and cattle. And my pet, it's still, we believe that animals have moral rights, and it's ethical, it's cruelty to animals to allow what we just talked about. Just because clones are genetically, I could say identical twins too, are genetically identical, it does not mean that they'll look exactly, their genes won't necessarily be expressed in the same way. So they could vary physically. I mean, they can look slightly different because different genes get turned on or off by the environment that DNA is in. And of course, behaviorally, I mean, your environment shapes how you form psychologically. So clones aren't necessarily going to think and act the same way as each other, especially if they're in different environments. Even when they're in a surrogate mother's womb, the uterus, different environmental factors in the womb can affect how that various aspects of that clone's development. And of course, the society that they grow up, the nutrients they get, the education or lack of it, that all affects what the embryo turns out to be. Gene therapy is a way of adding healthy genes into an organism that has defective genes. So let's say you're missing the genes or you have damaged genes that give you cystic fibrosis. It'd be great if we could add the right genes to the embryo, the fetus, so that that person doesn't have to suffer when she or he gets older with this cystic fibrosis, because they have the normal genes now that will fix the issue that the defective genes can't. So what we need to do for gene therapy is to somehow get into the cells that are missing, that have defective genes, injecting these genes into those cells enough that it'll make a difference. So we can do it into skin cells, blood cells, bone marrow cells. And this is currently what researchers are trying to do, especially to fetuses, so that the baby can be born with genes that will make up for the defective genes by making the protein that the defective genes can't. Now, when you do that into the somatic cells, transfer the proper genes into somatic cells, that is not going to be passed on to the person's sperm or egg into their children. So there's ethical issues of us changing the genes in the egg or sperm, and of course we can't do that anyway, so no one's trying that right now. So you can have gene therapy even if you are already born. Let's say you here in this example we've inserted the normal human gene into the plasmid of a bacteria. Okay, we let that bacteria culture over and over. We extract the bacteria plasmid, and we break off that normal human gene, and we package it in a virus, in like a code, or some respiratory virus. So we package it in there, then we let the patient inhale that virus, and it gets into his lungs, into the air sacs. The virus then invades the cells of the air sacs, and its human gene gets inserted into the nucleus of the lung cells. And so those cells, when they reproduce, they'll have the normal functioning gene that the person with cystic fibrosis doesn't have. Now, if you look up the success of this kind of procedure, it's really not super successful yet. There's a small improvement in the lung tissue breathing with this therapy, but the person still is gonna have mostly a condition that will eventually kill them. So we're still working out the bugs of this. Now, there's a problem. Once we get good at gene therapy, it'll probably be very, very expensive, and only wealthy people, very wealthy people, can afford it. So we could ask another ethical issue with gene therapy is, to what extent will we use it? Can I have designer genes that make my child be what I think is prettier than, or more handsome, or stronger, or a better athlete, or smarter? Do I have the right of choosing what I think is good for my child to be? And who says what we think is good is really good in the long run for the population? So am I playing God too much for these cosmetic? It's like a cosmetic surgeon I don't like the way God gave me this nose. Can I have a surgeon fix it? Now, in a lot of those cases, it goes bad, and you look worse after the cosmetic surgery. So, is it worth the risk? And anytime there's gene therapy, there's a risk during the surgery, during the procedure. And is it fair to a development baby to stress them out like that and cause possible risk for death? So a lot of the crops that we have today, important food crops like corn, I'm just going to pick corn for example, these crops have evolved over time by a process called artificial selection, where we've selected the species of plants that give us a better, more nutritional, satisfying fruit than what was originally naturally selected for, that naturally evolved. So here's an ancestor of corn. It's still out there, but look, how nutritious is that? Not very much so. And same with banana, watermelon, yuck, all kinds of things. This was carrots. So what we did was we picked the mutant forms, the variation, and we didn't grow the other forms in our fields. So we picked the weird ones that cropped up that had more nutritional value, and we planted those. And again, we got rid of the less nutritional ones in our agricultural fields, in our farms. And eventually we got the corn, what we have today. We got the watermelon that we have today, bananas that we have today. These primitive forms still exist. I mean, these naturally evolved forms still exist, but we don't farm them. So it took centuries to do this. There wouldn't be the cows we have now with huge udders to produce milk if there wasn't artificial selection by man to pick the cows that had bigger milk glands. We wouldn't have the crops we have now with a lot of fruit on the stalk instead of the little amount that are in the natural ancestral crops. There's now a new method that speeds up artificial selection to make more nutritious crops. And that's to use genetic engineering to make what we call GMO, or genetically modified organisms, which are transgenic organisms. So basically, we just we take a gene from another plant, we insert it into a plant we already have instead of DNA, and we get recombinant DNA, causing that new plant to form maybe a better protein, a more nutritious protein, or maybe making it more resistant to pests, or making it more drought resistant. So first step making a GMO is to find another plant. Oh, this would be good. This trait would be good in my crop. So you got to identify that trait and do a lot of experimentation to see how you can isolate that gene that you want to insert into another. Then you have to come up with a process to isolate that gene, using restriction enzymes and finding a transfer mechanism to get it into the plant cell of your crops or your flower that you want prettier or more durable. Then you have to grow those plants and then do tests to see if there's any bad result from you having put that gene into that organism. So that might require animal testing, animals eating that stuff and seeing if they get sick. No, big problem with testing. Sometimes it doesn't catch certain things, like especially allergens, things that the plant might make. Something after you put the gene in it, the plant might make some people that eat it really allergic to it. Not only that, but you gotta grow it out in the wild to see how it affects the ecology of the place. You don't want it to damage any part of the ecosystem. For example, its pollen could be transferred to other plants and cause those other plants to not grow correctly or to overtake, to gain something that gives them an advantage so that they overtake the crop that you made a GMO. A lot of times these kind of things are unpredictable. That's why you have to grow them. You gotta experiment in the field with them for years before you can say, okay, it's good to go. Then you have to sell it to the farmers. Usually farmers will want them because they have higher yields. You get more fruit per area of farm. The fruit is better quality. Maybe it'll last longer. It won't bruise as easily if it's like fruit, like tree fruit, when it's transported. Maybe it's allowing the farmer to use less fertilizer because it's more efficient. So, but of course, the cost of making a GMO will be transferred to the farmer, and he or she would have to pay more money for the seeds of GMO plants than of non-GMO plants. So let's look at some risk that occurred with GMOs. Even after testing, not enough tests were made. And that's why these days we're more thorough on our testing. So these potatoes that were GMO'd for something, when rats were fed them, they got a higher incidence of cancer and immune system issues as well. So lot of people are allergic to soy. The GMO soy produced a chemical that a lot of people were found to be allergic to. Animal tests on rats showed these kind of problems. Now, these allergies and problems were caused by genetic engineering, engineers putting a bacterial gene into the soy gene, inserting that, and causing it to be resistant to an herbicide called Roundup. The beauty of this idea was that you could spray the crops with Roundup, and this would kill any weeds that were growing up with the seeds of the crop. The soy plants would be unaffected. Well, that caused a lot of allergies in people. So here again is going back to the rat studies, where you had a high incidence of infant mortality in rats that had GM soy, something in the Roundup gene. The Roundup resistant gene was causing that. And same here, you had growth issues even if they survived. GM soy groups stunted the growth. So another great insecticide is BT. Bacillus thuringiensis. That's the scientific name for the soil bacteria that makes a poison that kills insects that eat that the bacteria gets in. Okay, we have used we have put the bacteria in water and sprayed it on crops for the last 50 years because it keeps insects that try to eat the crops from surviving. It kills them. Now what we do is, now that we got genetic engineering, instead of spraying the crops, we do a more efficient way. Because when you spray the crops, when it rains, it washes off into the soil. And so insects then can eat the crops again if the BT toxin isn't on it. So what they do now is they insert it in the seeds so that the BT won't wash off in the rain. And so when the plant grows up, in the seed is the embryo of the plant, and you put it in the seed, it incorporates into the cells of the embryo, and then when it grows up, all the cells have the BT in them. And so this would kill the insects that were eating the crops even after many rains. And it also protected the roots, which weren't protected from the spray, because when you spray the plant, it didn't cover their roots. So here we go again with how the BT gene was inserted into the seeds. So basically, you're taking the gene out of the bacteria that produces that toxin using restriction enzymes. And then you put it into another bacteria that has the ability of entering the plant, that particular crop plant's cells. And those cells are growing on a culture plate. Those are the plant cells. And then the bacteria has been absorbed by those. Then you grow those into plants. Those plant cell tissues really is tissues. And you grow them into plants, and each of the cells in that plant has the bacteria with the inserted gene from the Bt bacteria. I guess Bt doesn't insert itself, so you need a secondary bacteria that does. The Bt bacteria doesn't invade plant cells, so you put it into bacteria that does. Okay, so then you let those produce seeds. You grow those outside. You let them produce seeds, and then you sell those seeds to the farmers. There was a problem though. Monarch butterflies will die after eating Bt. So the corn pollen gets on milkweed plants in fields nearby. Milkweed plants near the corn fields. And what happens is the milkweed incorporates those bacteria from the pollen, and when the monarch butterflies eat that milkweed plant, which are specific, that's their primary food source, those monarch butterflies die. Also, some people are very allergic to the proteins from the Bt in the GMO corn. So you don't have a silver bullet, a magic bullet that fixes everything. There's always some side effect to unpredicted side effect. And that's why even with the most careful research, you're still going to have these things cropping up. So just review good things about DNA technology. We can use bacteria by splicing human genes into the bacteria to make drugs for us that were previously very expensive to synthesize. We can make effective vaccines. For example, the COVID vaccine was made by isolating the genes in the COVID virus. Once the genes were isolated, we could make that gene and change it so it wasn't going to cause COVID symptoms. And then we injected that into people, that's the vaccine, before they got COVID, so that their white blood cells, their immune system, could learn that what that gene was, so that when it did attack, the white blood cells would have already been educated and ready to pounce on it and kill it before it got out of hand in the body. So, in agriculture, we can transfer plants we can transfer genes from bacteria that kills insects that eat the plant. And so we can use it for built-in insecticide. As we mentioned with BT, then you can use it in crops to protect them from weed killers, herbicides. So it can do herbicide-resistant herbicides are weed killers. Not just weeds, any plants that are growing fast. Herbicide resistance genes can be inserted, such as Roundup resistance. And then, I didn't mention this, but you can insert the genes that are in nitrogen-fixing bacteria. These are bacteria that grow in the soil and provide nitrogen for plants. You can put those genes directly into the crops. So you can put fertilizers, built-in fertilizer. You won't need fertilizer because you're making a plant be able to take the nitrogen out of the air and turn it into the nitrogen needed by the plant to make its proteins.